Welcome once again to the new session of Premade Pro. We are continuing with the molecular basis of inheritance, and we will today start from the DNA replication. Okay, the exam form fill up has already been started, and we need to pace up the matter because we need to complete the syllabus by 1 month. Okay, so I will start with the DNA replication. I will teach those topics in which are very important for this examination, and the question will come from these topics only. Okay, so we come to the DNA replication. DNA replication. How the DNA is replicated. Okay, this is the chapter where I need to draw many of figures, and I need to explain this after drawing the figures. So keep patience and see the matter. Okay, so let's start with drawing the picture. Okay, so let me draw it. Don't ask questions in between. Okay, I will definitely give the explanations. Now this is the 5' end, this is the 3' end, and this is the 5' end, and this is the 3' end. So what is it? Okay, let me consider it as A, this is B, this is C, and this is D. I am giving the index here of this, and let it be the index. What is A, B, C, D? Upper, first one is first and last. The first one is A, the last one is B, and in the lower segment the last one is C and the first one is D. Okay, so now this is be the origin of replication site, that is the ORI site. Let this is be the ORI site, this is be the ORI site. ORI means that is the origin of replication. Origin of replication. Okay, origin of replication. Now the main enzyme that is required for the DNA replication is DNA polymerase 3. Okay, the main enzyme DNA polymerase 3, it is the main enzyme. DNA polymerase 3 is the main enzyme. Okay, and DNA polymerase 1, it is called the Kornberg enzyme. It is called Kornberg enzyme. Okay, now what happens? What is the base pair per second of the DNA polymerase 3? It is 2000 base pair per second. This question comes in the medical exams, and these questions are generally overlooked by the candidates. So base pair DNA poly 3 is 2000 base pair per second. Okay, now here an enzyme that is required to unwind the DNA strands or open the DNA strands, it is done by the helicase enzyme. Helicase. unwinds the DNA. Unwinds the DNA. Okay. I am writing a notes here. Okay. Some important points, topics. So let me write helicase. Helicase another name is called unwindase. These names are changed to just to haphazard you in the examination, because at the examination moment, at the examination hall, all the candidates are a bit of in stress. So to check your stress holding capability, this type of name changing comes into the action. Okay, this function is to break the bonds. Breaks. Okay, next H2 bond. H2 bond, that is the denaturation. Denaturation oblique unwinding of DNA, oblique unwinding of DNA. Denaturation or unwinding of DNA. Now it comes to replication fork. Replication fork I will all discuss after writing it. I will discuss every topic. Replication fork. The next action is of ORI site. ORI site. What is the function of the ORI site? Its function is to start the replication. Origin of replication. Here the replication starts. Origin of replication. The next one is, this is the actual site, atric site, easy to break. Atric site is the point where the replication starts. This you will not find in any book. Okay, atric site, easy to break. So from this side the replication starts. Okay, double bond compared to triple bond. Triple bond, the number of bond, as the number of bond increases, the energy to break the bond also increases. So a single bond is easier to break than the double bond. The double bond is easier to break than the triple bond. So it will start from A double bond T site rather than from G triple bond C site. Okay, rather than C. Okay. Next one is DNA unwind occur at regular interval of time. जहां जहां मर्जी वहां नहीं होता है, जहां होना चाहिए वही होता है. ठीक है, at regular interval of time. So and never unwind whole DNA at the single strand binding protein. whole DNA कभी एक साथ unwind नहीं होता है at time. Okay, the next one is the SSB. SSB means a single stranded binding protein. Single stranded binding protein. The single stranded binding protein prevent the renaturation of the DNA. Prevents the renaturation. Denaturation and renaturation. Denaturation means breaking of the DNA, and renaturation means the joining of the DNA, but renaturation of DNA. Okay, now the next one is due to helicase activity, supercooling occur. prevented by topoisomerase. This is prevented by the topoisomerase. It is prevented by topoisomerase. It is an enzyme, isomerase, prevented, and DNA guidase. Topoisomerase and DNA guidase. Okay. Now, what happens? Let me draw another figure. See what I am drawing. Just you need to understand this, because it can generate many number of questions. Okay, it can generate many number of questions. Okay, now now see, this is the 5' end, this is the 3' end, this is the 5' end, and this is the 3' end. Where this is the 5' end, so this is be the 3' end. Okay, now this is the 5' end, this is the 3' end, and this is the 5' end, this is the 3' end. Okay, now what happens, this here it is CMP oblique GMP, cytosine monophosphate and guanosine monophosphate. Here it is of CMP and GMP there. Now it is broken by, it is the phosphodiester bonds are being broken, breaking of phosphodiester bond. Cut DNA, tension release, joined. Okay, now what happens, this is OH, this is the 3' OH. Now here it is a leading strand towards replication fork, Okay, now the DNA, here it is bond, the hydrogen bond is here. Here the hydrogen bond is there. Okay, now the DNA synthesis, okay, the DNA synthesis goes from 5 prime to 3 prime end. You all know it. The DNA is anti-parallel. RNA primer synthesis is done by primase enzyme. RNA primer synthesis is performed by the primase enzyme. Synthesis by primase enzyme. Okay. To initiate the DNA replication, to initiate the DNA replication, we provide the 3 prime OH site provides the group for binding. Provides the group for binding first nucleotide of the DNA. Okay. Now what happens? The DNA polymerase does the DNA synthesis. Okay, from the 5 prime to 3 prime synthesis, polymerization activity. Okay, in prokaryotes, DNA polymerase 1, DNA poly 1, DNA poly 2, DNA poly 3. In prokaryotes, these 3 enzymes performs the DNA replication. Okay. Now the unwinding process. So unwinding process, again I need to draw picture. This chapter contains so much pictures, many pictures are there. Okay. Okay. Now okay. Now this is the this is the 5 prime end, this is the 3 prime end, and this is the unwinding. this is the 5 prime, this is 3 prime, and this is the unwinding. Okay, now the 5 prime oblique 3 prime, 5 prime to 3 prime, this strand 5 prime to 3 prime, this strand is called the template strand. Okay, oblique new strand, template strand. Okay. Got it? And the new strand here 5 prime to 3 prime, this strand is called, it is also a new strand, template oblique DID strand. Okay, now DNA is 50%, DNA is semi discontinuous. It is semi discontinuous. So we find the break of the continuity of the chain, so it's called a semi discontinuous. Okay, now why it is called semi discontinuous? Because 50% of the total DNA is of new strand and 50% is of the parent strand. Okay, so 50% DNA old oblique parent, 50% DNA new oblique semi conservative. Okay, got it? Okay, now what happens? Now this strand, let it be this strand, we name it as, this strand is 5 prime and this is 3 prime. Well, this is 3 prime and this is gonna be 5 prime. Okay, now this is the continuous, this is a continuous strand. This is continuous fashion. This is a continuous fashion. Okay. And the other strand is discontinuous in fashion. Okay. The 5 prime to 3 prime, it is here. Here it is called, this fragment is called 3 prime OH. It is called Okazaki fragment. So, and this is this is 5 prime, this is 3 prime, this is 5 prime and this is 3 prime. Okay, this is called the Okazaki fragment. Now the continuous strand, the template the template direction is always from the 3 prime to 5 prime, while the continuous fashion, the neat primer leading strand is 5 prime to 3 prime. So the template strand is 3 prime to 5 prime and the leading strand is 5 prime to 3 prime. Okay, the need for more than one primer. What is the need? Why there is a need for more than one primer? Need for more than one primer. Need for more than one primer is formed in form of small segment Okazaki fragment. Okay, and this is called the lagging strand. Okay, lagging strand is 5 prime to 3 prime. So lagging strand is what as the leading strand. Okay, and the template direction is 5 prime to 3 prime here. Okay, so from tomorrow we will start with the climax, how the replication completes, and then after the climax I will end up with the DNA replication with why it is called a bidirectional replication. I'll end up with that. Okay, and the replica on the segment of the DNA will be taught in the next lecture, because this lecture will be, if I teach in this lecture, it will be of 1 hour 5 minutes. Okay, so this is all for today. So please go through it and please read the NCERT of this chapter. Okay, so good night, and please share, subscribe, and like this channel, and please share it with your friends. Okay, thank you.